As Charlotte introduced, I'm actually a chemist by training, but my talk today is going to focus on the use of microorganisms for chemical synthesis. As was just introduced, I think this offers really exciting potential for the chemical industry to move away from its use of fossil resources as we all try and strive towards our net zero commitments. So just to start off, I'm going to start off with my glass half full introduction as well. Despite perhaps lots of negative press at the moment, and actually despite the need for many of the technologies that I'm going to present to you today, I strongly believe that the petrochemical industry is actually one of the greatest achievements that our society has developed over the past 150 years, where we have identified fossil fuels as a really reliable and abundant source of carbon. And we've developed really efficient and very elegant strategies to extract that feedstock and to transform it into many of the compounds or products that we rely on in our everyday lives. To give you just a few examples, such as compounds to treat cancer, compounds to treat anti-infection or infectious diseases, to the humble paracetamol. These are all compounds that are derived from fossil resources currently. Included in that are compounds in many materials that you're probably wearing right now, and in many monomeric materials that can be polymerized into products that we build our homes with and we sort of use in our everyday lives as well. And this also includes personal care products that you're probably again wearing right now, things like perfumes, cosmetics, shampoos, etc. However, I think we are genuinely at a bit of an impasse in the way that we conduct ourselves industrially at the moment. And the overarching reason for that is that this industry relies entirely on the use of what is a fundamentally unsustainable natural resource. So fossil fuels are reducing, and they are predicted to run out. In a number of years, it's quite highly contested in the literature, but I think it's widely agreed that that number is a 2-digit number, and it's not a 3-digit number. So this is going to happen in the lifetimes of my children and my grandchildren, and therefore we need to be developing technologies now if we are to move ourselves away from our reliance on this unsustainable resource. And the reason that this is so problematic is that we consume at present over 10 billion tons of oil every year for use in fuels and in the manufacture of chemicals. And despite many of the sustainability initiatives that have been around for the past 20 years, most industries are reducing their reliance on fossil fuels, but the chemical industry is the only sector whose reliance on fossil fuels is currently increasing. And this isn't just a practical problem. The effects of both these industrial activities are having an irreparable impact on the planet around us. Since we started commercializing industrial rubber production in the mid-1960s, 20 chemical companies have released over 480 trillion tons of carbon dioxide into the atmosphere. And these industrial greenhouse gas emissions are a leading driver, to most people, believe this is a leading driver of global climate change. And in addition to that, the waste that is generated from these industrial activities is polluting ecosystems worldwide, and this is having a dramatic effect on global biodiversity. And as I mentioned, despite our best efforts, you can see here, this is just a graph showing atmospheric carbon dioxide emissions from industrial activity since the mid-1970s to the present day. And you can see we've never really been able to move away from this upward trajectory in our release of CO2. And actually, from the time I was born in the late 1980s until now, atmospheric CO2 emissions have doubled. However, it's not all doom and gloom. You can see this sharp spike here, and that was in 2021. We've seen a sharp decline in atmospheric CO2 emissions, and that was, of course, due to reduced activities and reduced travel as a function of the COVID-19 pandemic. So if you're to take any positives from what's happened over the last couple of years, be it that sharp changes in the way that we conduct ourselves as a society, the way that we manufacture, and the way that we travel can have positive benefits to the carbon that we emit into the atmosphere. So I'm going to talk to you today about microorganisms. And as I tell everyone who enters my lab, microbes are nature's synthetic chemists. They are incredibly highly evolved chemical synthesis machines that can use a diverse range of sustainable feedstocks and can transform these into a huge array of industrial products. And just to give you some examples of the sorts of chemistry that microorganisms can do that is quite remarkable. So Anammox bacteria, such as shown here is in this Candidatus strain, can fix molecular nitrogen from the atmosphere and turn that into ammonia. This is a process that we rely on petrochemically currently to produce fertilizers. This is a native mechanism present in endogenous bacteria. Clostridial bacteria can fix carbon dioxide from waste industrial processes and carbon dioxide actually straight from the atmosphere and turn these into a variety of platform chemicals. Again, this is something that we conduct petrochemically from fossil resources currently. And perhaps a bit more colloquially, Pseudomonas syringae is a beautiful bacteria that actually has this remarkable capability to freeze water at room temperature. And this process has actually been commercialized currently for those of you who enjoy indoor skiing. The snow that is produced in an indoor skiing rink, is that right? No? Slope, yeah, is actually produced via a microbial process. And we can utilize industrially not just the native capabilities of microorganisms, we can design them or engineer them to do new things and make new chemicals. And this is something we're particularly adept at doing here in Edinburgh, where we have access to an ever-growing library of DNA sequencing data. We can effectively read DNA faster than ever before. We can write DNA faster than ever before. And we can translate this faster, more quickly into industrial bioprocesses than ever before. And particularly in the School of Biological Sciences, we have a facility called the Edinburgh Genome Foundry, which is one of the most fully automated DNA assembly facilities in the world, where we can effectively build new microorganisms very, very quickly to perform new functions and produce new products. And actually the area that I'm particularly interested in and is going to be the focus of my talk today is actually the utilization of industrial waste and its biotransformation into industrial products. You'll be quite glad, especially before dinner tonight, that I'm not going to talk to you about sewage waste, but it's great to see our industrial collaborator here today. And I think this is a really, conceptually, a very interesting approach for industrial biotechnology because it achieves two things at once. It takes a waste feedstock, i.e. a source of carbon that would otherwise be sent to landfill or incinerated, and it re-enters that carbon back into the national chemicals economy. And at the same time, you actually use that carbon to generate a product that would itself be manufactured from fossil fuels. So you're both reducing waste and you're diminishing our reliance on fossil fuels at the same time. So I think the upcycling of waste using engineered microorganisms is going to be a really promising and very, very powerful approach in the manufacture of sustainable chemicals in the future. So the first waste source that I'm going to talk to you about today is actually lignin. So lignin is a very highly abundant natural resource. It's widely thought of to be one of the most untapped sources of carbon on Earth. It's also generated in over 70 million tons every year as a byproduct of the agriculture and paper milling industries worldwide. And interestingly, the major component of depolymerized lignin is this molecule here. This is called guaiacol. And at the same time, guaiacol is actually a microbial metabolite. So it can be engaged in microbial pathways and transforms into new products. And the target compound that I'm going to tell you about is this molecule here called adipic acid. Adipic acid is probably the most simple compound I've ever worked on in my career, but it's arguably the most important. If you've never heard of adipic acid, you're most likely in contact with it right now because it's used in amongst its many applications in the manufacture of nylon 6,6 polymer for use in clothing. We rely on adipic acid quite ubiquitously throughout our modern day lives, and as such, we have a huge demand for adipic acid that requires almost 3 million tons to be produced annually. And this is done entirely from petrochemical-based feedstocks, in fact, benzene. Not only does this rely on a fundamentally unsustainable natural resource, the manufacturing route that we use to get to adipic acid releases a mole of nitrous oxide. And nitrous oxide is a greenhouse gas that's over 20 times more potent than carbon dioxide. And actually, a couple of years ago, it was documented that almost 10% of all human-associated nitrous oxide emissions worldwide come from this one industrial process. And to put that in a bit more context, the environmental benefits of replacing this process with a biotechnological equivalent would have the same environmental benefits as electrifying all the cars in the UK twice over. And again, adipic acid is a microbial metabolite. So we thought there was a really interesting opportunity here to valorize waste into adipic acid using an engineered microorganism. And to cut 3 years into one sentence, that's in essence what we did. So we combined 4 enzymes from throughout nature, from various bacterial sources, Amalcolitopsis, Pseudomonas butida, and Bacillus coagulans, which is a derivative of the bacteria you find at the bottom of your shower. And we encoded all of these enzymes onto a single bacterial overexpression plasmid. This is effectively a circular piece of DNA that tells the cell to perform new chemical reactions. And what we were able to show is that we could take guaiacol derived from lignin waste, we could demethylate this to give you catechol. This could then be oxidatively cleaved to produce cis-muconic acids, which then could be reduced to form adipic acids. Critically, this process is very efficient. We produce adipic acid in over 90% yield currently. It's a one-pot process that happens in water. It happens at room temperature. And critically, it doesn't release any nitrous oxide at all. The only byproduct from this process is water. And we published this a couple of years ago now in ACS Synthetic Biology, and we're really grateful to have recently been awarded an EPSRC Sustainable Manufacturing Grant to pioneers, maybe a bit too self-congratulatory, to intensify this process and assess its commercial viability with a number of industrial partners from 5 different countries. And this will be starting actually at the University of Edinburgh this year. So the second waste feedstock that I'd like to talk to you about today is plastic waste, and we've heard a great introduction to this from Jenny earlier. And I don't have to profess to everyone here the severity of the global plastic waste crisis and the fact that we really need to reassess our use and disposal of this industrial product. I was really fortunate to be involved in COP26 at Glasgow at the end of last year, and somebody presented to me there that if we were to take all of the plastic wastes present in the world right now, we could create a plastic bag that would bag the Earth. I think that's a really nice way to think about it. It's not nice at all, but it's a way to think about it. And the ubiquity of this material in our everyday lives means, again, that we have to manufacture quite a lot of it. We currently manufacture over 30 million tons of PET each year. And the problem with this is that 80% of this PET that we manufacture is designed to be single-use. And although recycling technologies are being developed and will no doubt continue to become more and more efficient, when we recycle plastic into second and third generation materials currently, these materials are of lesser value than the original virgin polymer. And therefore, we enter into this downcycling approach where we produce less and less valuable materials that ultimately just end up in the same place as they would do in the first place. So they generate the same waste. Now, this was a project that was pioneered by a great postdoc in my lab and now colleague of mine in the School of Biological Sciences, Joe Sadler. And what Joe realized is that the depolymerization product of PET plastic is this molecule here called terephthalic acid. And again, terephthalic acid is a microbial metabolite. And therefore, we thought we could transform terephthalic acid using engineered microorganisms into something more valuable, therefore avoiding this downcycling process into less valuable and ultimately waste. And the molecule that we focused on very recently is this molecule here. It's vanillin, which is the main component of vanilla flavoring that's present in foods, in many cosmetics as well. And 80% of global vanillin production is produced currently from fossil resources. So therefore, as terephthalic acid is a metabolite in nature, it can be a substrate for synthetic biology. Can we access vanillin from terephthalic acid using a single engineered microbe? And again, that is in essence what we did. This is a slightly more complicated pathway. In fact, there's 3 enzymes involved in this first step here. So there's 6 enzymes involved in this pathway in E. coli, where we, in essence, use molecular oxygen to oxidize terephthalic acid into this intermediate here, which is then processed via this aldehyde here, eventually to vanillin. The chemistry doesn't really matter in this, at this stage. Nevertheless, these enzymes were isolated from Comomonas bacteria, from Nocardia bacteria, and from the common household rat as well. So this process, again, we managed to achieve 79% conversion of vanillin directly from terephthalic acid in a single flask bioprocess that occurs at room temperature, again, at ambient pH, and produces vanillin in quite high conversions. But probably the most exciting thing that happened in this project was that Jo, on her way to work one morning, came across a plastic bottle on the streets of Edinburgh. We took this into the lab, we punched out a hole in this plastic bottle, we depolymerized it, and we simply fed it to our bacterium, and we found that we were able to efficiently convert this into vanillin in the lab. And this was the first, to our knowledge, example of where somebody had taken post-consumer plastic waste and converted it using an engineered microbe into a higher value industrial product. However, to me anyway, the most exciting thing about this project is not that we can make vanillin. It's the fact that when we do make vanillin, we make very little of anything else. And what that tells me is that we have a really efficient process to take terephthalic acid from plastic waste and convert it into this molecule here. This molecule is called protocatechate, and it's a central metabolic intermediate in many microbial pathways. And the reason this is really exciting to me is because protocatechate serves as a substrate for many, many bacterial pathways that will enable us to access a huge variety of industrial products, from other cosmetics to materials, and more excitingly, to pharmaceuticals. And in essence, what we think this is enabling us to do is to take a waste stream that is potentially, is damaging the environment worldwide, and to use industrial biotechnology to transform that into something that can potentially benefit human health. And this is something we're working on actively at the moment in Edinburgh, and hopefully I'll be able to present this to you in maybe next time. Okay, so all that leaves for me to do is really to thank the incredibly talented group of scientists that I get to work with every day. I've highlighted a couple here that have been specifically involved in the work that I presented to you today. Jack is a PhD student who has almost single-handedly developed the adipic acid pathway in E. coli, and he is funded by the Carnegie Trust. Jo is a postdoc, now colleague, now Chancellor's Fellow in the School of Biology, who pioneered the vanillin pathway in E. coli. I'd like to thank our funders, specifically UKRI and the University of Edinburgh. All of our industrial collaborators, I have to thank you. You really are what inspires our work here at the university. And really just to give a shout out to two more projects that our students are working on currently. For those of you with an interest in hydrogen, Miriam is currently pioneering a pathway to hydrogen in E. coli, aiming at sort of replacing petrochemical-based steam reforming methods to produce hydrogen. You can see some of the strains here are pretty effective. They're actually bulging off the top of the bung on the flask. And we're working with our colleagues, Louise Horsfall in the School of Biology, to harness this pathway in D-sulfur vibrio to produce palladium catalysts. And we've shown recently that these are highly efficient chemical catalysts in many cross-coupling reactions that are used in the pharmaceutical industry. And then finally, I'd just like to give a quick mention to the Science for Sustainability Hub. This is a network of researchers from across Edinburgh University with a vested interest in sustainable research. It was launched in June this year, and its aim is to build partnerships across industry, NGOs, and civil society. It aligns directly with UN Sustainable Development Goals, and its aim is really to put us in a very strong position to react very quickly to upcoming funding calls in the area of sustainable research. I direct you to this website here at the top if you'd like to register your interest or join us in the Sustainability Hub. Our lab are part of this and actively engaged in their activities, and I've given you a sort of an outline, I guess, of some of the other activities and interests in this area that we have here at the university. Thank you. I'm happy to take any questions.